Good morning, everybody. In last lecture, we have discussed regarding neuroregulation of respiration. Just to remind, we have learned that there are three centers which regulate the respirations located in the brainstem, that is pons and medulla. The first and foremost and vital center is called as DRG, dorsal respiratory group of neuron. Other one, VRG, that is ventral respiratory group of neuron. Third one, that is pneumotactic center. We have learned that from DRG, ramp signals are arising for a period of 2 seconds. They are ending on the motor neurons which are supplying the respiratory muscles. That is the phrenic nerve which is supplying the diaphragm, and intercostal nerves which are supplying the external intercostal muscles. When impulse travels from DRG, they will stimulate the respiratory muscles for a period of 2 seconds. The diaphragm will contract. It descends. There is increase in vertical diameter of lungs. The intercostal muscles contracts, that is external intercostal. It lifts the rib cage up, that is called as bucket handle movement, leading to increase in transverse diameter of thorax cavity. Both of this increases the lung volume. With Boyle's law, with increase in volume there is decrease in pressure inside the lung. Air is sucked inside, that we call as inspiration. Then DRG takes a pause for 3 seconds. The muscles relaxes back. Diaphragm and external intercostal muscles come back to original position. Volume decreases. Pressure comes back to normal. Air is expelled out, that we label as expiration. So normal quiet breathing is controlled by DRG. We learned that VRG also supplies the same neurons, but it is inactive during quiet breathing. But it comes to action when we need extra breathing, where our accessory muscles of respiration come into action, both for inspiration as well as expiration. So they are helpful in forced breathing. And this you can see that from DRG impulse is traveling down, it is supplying to the muscles of respiration. Then now pneumotactic center is acting as a brake only when there is inflation beyond certain limit. We will be learning this, their function during mechanical control of respiration. Okay. Now these are the accessory muscles of respiration. You can see on the right side they are moving upwards, that is they will lift the rib cage upwards. These are sternocleidomastoid, anterior serrator, splenic. This will lead to forceful inspiration. On the left side you can see internal intercostal muscle, direction is downwards, that is they will pull the rib cage down forcefully. Internal oblique, external oblique, transverse abdominis muscle. So these are called as accessory muscles of expiration. So this ventral respiratory group of neurons, VRG, you can see impulse is traveling from there to the respiratory neurons which are supplying the respiratory muscles. From here it is going to both sides. You can say this will cause the inspiration. This side will cause the expiration. So it is forceful inspiration, forceful expiration when needed. Okay. So they are inactive during normal quiet respiration. Electrical stimulation of few neurons causes inspiration, whereas other causes expiration. This area operates more or less as an overdrive mechanism when high levels of pulmonary ventilation are required. This was our discussion in the last lecture. Now, these neural centers are affected by, first, central chemoreceptors, Then sensory termination of afferent nerves form peripheral chemoreceptors, baroreceptors, and several types of afferent from the lungs and from the body. Okay? I will see first the central chemoreceptor. These are located again at the lower end of brainstem, that is at the medulla. Why they are called as chemoreceptor? Reason is that these are central chemoreceptors located at the lower end of brainstem. They are being influenced by our respiratory neurons, which are located in pons and medulla. You can see from both. The central chemoreceptor, they are called as medullary chemoreceptors simply because they are located in the medulla. H-ions, the only important direct stimulus for these neurons. But H-ions penetrate the blood-brain barrier slowly. The central chemoreceptors, you can see this is the blood-brain barrier, but we have got alternatives how they will get stimulated. You can see in the blood we are having carbon dioxide. So if there is increase in the PCO2, it will enter in the blood-brain barrier. CO2 combines with water to form H2CO3, which dissociate to form H-ions and HCO3. This H-ions has got influence on central chemoreceptor. So indirectly CO2 will influence the central chemoreceptor through H-ions. H-ions penetrate slowly, but CO2 can enter the blood-brain barrier quite easily. So increase in PCO2 level will stimulate the respiration indirectly through H-ions. So that's why CO2 is very, very important. You might have learned breaking point in the respiration. So the same thing. Whenever we are holding the breath, gradually as we are not expiring it out, CO2 accumulates in the blood. Up to a certain level, CO2 will enter the CSF. There is increase in H-ions indirectly, leading to strong stimulation of inspiration, and it cannot hold the breath beyond certain limit. That is called as the breaking point. So breaking point depends on the CO2 level in the blood. So these are the respiratory centers, central chemoreceptors, influence. So they are stimulating the respiratory centers, which will now again come back as the mechanism we have learned. From DRG, impulse will come down to our neurons located in the spinal cord, that is phrenic nerve neuron and intercostal neurons, which will again go back and there is a respiration. So chemoreceptors are again indirectly stimulating respiration through neurons. So they are influencing again back to the neurons located in the brainstem. Okay. So CO2 readily penetrates blood-brain barrier. CO2 is promptly hydrated. H2CO3 dissociates so that the local H-ion concentration rises. The H-ion concentration in the brain interstitial fluid parallels the arterial PCO2. Means increase in arterial PCO2 indirectly will increase H-ion concentration in the brain. Changes in oxygen concentration have virtually no direct effect on central chemoreceptors. Excitation of respiratory centers by CO2 is great the first few hours. It is very important in exercise. Gradually declines over the next 1 or 2 days, decreasing to about 1/5 the initial effect. Now see the peripheral chemoreceptor, as the name itself indicates, they are located peripherally in aortic bodies and carotid bodies. If you just recall your regulation of baroreceptor mechanism, they are located in aortic sinus and carotid sinus. Here we are having bodies, special receptors are there which will sense change in blood chemistry. Having a close look now, peripheral chemoreceptor system, aortic bodies. From here vagus nerve is starting. Carotid bodies taken by glossopharyngeal nerve. Both are ending in medulla. Here they are influencing down again our neurons which are regulating respiration. That is our DRG, dorsal respiratory group of neuron, our VRG, that is ventral respiratory group of neuron, pneumotactic center, okay, which are again converging down on the respiratory motor neurons. How there is stimulation of this aortic and carotid bodies, that is peripheral chemoreceptor. So these chemoreceptors, if you take a section of aortic or carotid bodies, these are called as glomus cells. Whether it is aortic body or carotid body, glomus cells. Now in that they are having channels called as O2 sensitive potassium channels. Okay? There is a efflux of potassium from inside to outside, and this is O2 sensitive. These positive ions are coming from inside to outside. You can see potassium is coming from inside to outside. These are glomus cells. Okay, this is O2 sensitive. Now simple, if O2 level reduces, what can happen? This channel will close. Simple, because they are O2 sensitive. So if O2 oxygen level decreases in the blood because of any reasons which we have discussed, that is hypoxia, we have learned in detail what are the causes of hypoxia. Because of any reason, if there is decrease in oxygen level in the blood, this channel will close. What will happen? Positive ions remains inside. Potassium cannot come. That is called as depolarization. Again, you recall your physiology of action potential. Normally, inside is negative. Once inside becomes positive, it is called as depolarization. So here, potassium ions are not coming outside. So this potassium ion increases, positivity rises, that is depolarization. They are stimulated, impulse will travel down. So, peripheral chemoreceptor system detects changes in oxygen in the blood. If you recall, we have learned central chemoreceptors are sensitive to CO2 levels and H ions. More on H ions, enlarged on CO2. But peripheral chemoreceptor system detects changes in oxygen level in the blood, particularly sensitive to changes in arterial PCO2 in the range of 60 down to 30 millimeters of mercury. We have learned that PO2 range is normally 100, coming down up to 60. But if it is coming down below 60, they will stimulate our peripheral chemoreceptors. Each carotid and aortic body contains islands of 2 types of glomus cells. Respiration. If you just recall, we have learned central chemoreceptor, sensitive H ions and CO2. Peripheral chemoreceptor, they are sensitive to oxygen level and to a slight extent CO2 enhance. But ultimately both of them are influencing our neuronal centers. So ultimately influence again coming down from DRG. Now there are some non-chemical controls. Means there is no influence here of H ions, CO2 and oxygen. Non-chemical control means from vagal efferents from receptors in the airways and the lungs. Efferents from the pons, hypothalamus and limbic system. Simply you can see when you are emotional, your rate of respiration changes. So this is the influence coming from limbic system. Efferents from proprioceptors. A person starting for the running. The moment before this he starts to run, sprint runner, you can see rate of respiration rises. Why? Coming down to this from proprioceptors. Efferents from baroreceptors, arterial, atrial, ventricular, pulmonary. Okay? Now these are located in the lungs. These are called as stretch receptors of alveoli. Now here we will be taking one try. One of the Indian scientists have discovered these receptors located in the interstitial space of the lung. Anybody can tell? I will tell you, this is Dr. Painter's sir. He has discovered the J receptors. So these receptors are located in the this is called as Herring-Brewer's inflation reflex. Means whenever we are taking inspiration, after certain expansion, we cannot take inspiration beyond. Why? Because stretch receptors located in the interstitial space of the lungs, they get stimulated. Impulse will travel down from the vagus to go to again our neural centers of respiration, and they will inhibit them, and Ramsden stops contraction. Impulse will not travel down from there to the respiratory neurons, and diaphragm will not contract now. To stop contraction, expulsion starts. So it is called as Herring-Brewer inflation reflex. So stretch receptors located in the walls of bronchi, bronchioles throughout the lungs. When lungs becomes overly inflated, the stretch receptors activate an appropriate feedback response that switches off the inspiratory ramp. It is called as Herring-Brewer's expiration is stopped. Inspiration again starts. It's called as Herring-Brewer deflation reflex. Means when the lungs are expiring or deflated, beyond certain limits, will not let the lungs to get collapsed fully. Okay? So again impulse will come back to pneumote accidentally and stop. So that is Herring-Brewer's deflection reflex. Respiration. The body. Okay? So here you are finishing regulation